just the bounty here in Oregon. I forget. You know, I'm like, in Delaware, we grow pickling cucumbers and watermelons. That's about it. Maybe some lima beans, But here it's just like swath, like miles at the end of vineyards and filberts and all of these wonderful things. And bees are a really important part of that system. And in order to meet these pollination demands, because when you bring those hives into pollination, they have to be strong. They have to have a certain amount of frames of brood. They have to be disease-free. There's all these requirements. And we need to make sure our bees have enough food variety and nutritious pollen sources. That has to be the first priority. And in order to meet these pollination demands, sometimes we'll do other things to get them that strong, whatever it takes, because people's economic livelihood depends on it. But I'm trying to tell all beekeepers in my area, because where I live, it's mostly hobbyists and beginners, that nutritious food sources, that's the best you can do. If you're going to store your bees, the best thing for you is to provide them with a variety of nutritious pollen and nectar sources. Sometimes that's hard to figure out what those are.
There is nothing that tells us anything about the nutrition of pollen sources that I know of.
It's very, very limited, and I've scoured it. So if you want, we can talk, and I can give you a little bit of what I know.
Yeah, in New Zealand they've got it, but of course that doesn't cover a lot of California trees.
Yes, yeah.
Okay.
No, and absolutely, and also, you know, I recently saw some other work done where it covered a lot of different pollen sources, but then you have microclimate and variability even within the same tree, depending on where it's grown and what, you know, practices are used to grow that tree. So it's kind of, nothing's cut and dry. All right, so I'm going to go a little bit into the history because this is how I really got excited and really started my research, is just kind of exploring the history of humans and honeybees. And I often say I'm in a wildlife and entomology department, and it's an entomology and wildlife ecology department, actually. And I really like that combination because we're about conserving and understanding the ecological balance of the organisms we study. And I say that our early relationship with honeybees was much like humans and other wildlife. We hunted them, we used them for their resources. Oftentimes there was a spiritual connection with the hunt, with the harvest. And it goes back, it's very deep, it's very much traditional in our lore, our relationship with honeybees. You can see a honey hunter here. Of course, that's Apis dorsata, I believe, on a cliff. A lot more dangerous than maybe honey hunting or beelining might be, but you know, you can see him actually harvesting that. And actually, the history in North America, a lot of people still don't realize that we actually brought honeybees over here. That honeybees aren't native to North America. Though there is some, you know, people who like to debate that because in the Miocene, they found Apis nearctica, I believe it is, which was similar to a honeybee in Nevada. So there is some debate. But what we know of this type of honeybee that we have here today was not present in North America. And in the 1600s is our first record of them actually coming over. And this was via ship's logs. The first one that we found was from the Virginia Company. And also in that cargo were things like rabbits and apple trees and other things that were comfortable to these European settlers who were coming over to kind of settle in this new unknown world. And these and the resources they provided those people were really important. They didn't understand the pollination connection yet. It wasn't until the mid-1700s that they started making that connection between pollinators and insects and fruit and kind of that pollination connection. But over time, once they were brought in, we started being able to select for them. We started to actually understand, wow, there's a lot of diversity in our ecologies here, there's a lot of diversity in habitat, and people kind of decided what was important to them and be able to kind of select them for honey production, pollination, gentleness or temperament, and pest and pathogen resistance. And there's a really amazing beekeeping history here. I love these old kind of pictures. There's some steppe beekeeping there. Here's some bee gums here. And actually, if you go into different, more remote parts, when I was in North Carolina up in Appalachia, and probably remote part of every state, you can still find some more traditional beekeepers who do keep bee gums. It is illegal in a lot of states because apiary inspectors by law have to be able to inspect and remove combs to look for pests and pathogens in response to AFB quite a while ago. All right, and then after that initial importation event that we know, and there was probably others, that's just the only one we have, like documentation from the ship's log. I'm sure there was others, and there's anecdotal evidence to suggest that. But additional introductions occurred more than 200 years after this initial arrival. And we also know from that ship's log that it was the dark German bee, Apis mellifera mellifera, that was that early arrival of Apis mellifera. And about 200 years later, between 1859 and 1922, 7 additional subspecies were brought in. Now, the reason behind this was very different than when they first came over. It was kind of just an ag part of their livelihood and their farm. A beekeeping industry was starting. It was evolving, and they actually were bringing in, looking for strains that would overwinter well, that were good honey producers, that were more gentle. And if you look at the timeline back of some of the different subspecies they brought in, some of them only lasted for very short periods of time because they were nasty or because they didn't overwinter well. So this was a really exploratory time in terms of creating the basis of our stock here in the U.S. It's kind of exciting. So the occupation of beekeeping really evolved over time here in the U.S. to what it is today, this beekeeping industry. And you can't generalize it into any one thing. And that's what I'm realizing. When I lived on the West Coast, I was much more influenced by more large-scale commercial beekeeping because that's what was around me and what was needed. Being on the East Coast now in North Carolina and now in Delaware, I'm surrounded by beginners and hobbyist beekeepers who just like to sit in front of their entrance of their hive like this, and that's peace and happiness to them. So I kind of like that. It's nice. But really, the amazing thing about honeybees is their ability to thermoregulate their nest cavity. And that has allowed them to expand all over the place. They're much better at doing that than my air conditioner or my boiler, which always goes out in winter. They're designed. They're morphologically, physiologically designed to keep very tight control of their nest temperature and different other characteristics. And so that really has allowed us to provide them with a nest cavity and move them all over the place. We've expanded their range tremendously, which we'll look at in a minute. And also been able to select for favorable traits because we can bring them to different areas and then watch them in that area and see how they do and actually select ones, maybe have a few, you know how this goes, select ones that did well that winter and maybe make some queens off of her for the next season and so on and so forth. Also manage for honey, used for pollination. All right, so this is the actual range of Apis mellifera and also the rest of the genus. So Apis mellifera is just one species within the genus Apis. And what you're seeing here in the pink, this is all Apis mellifera, the species we know and love so well. And over there in the green is Apis cerana, which is the Indian honeybee, the true host for for Varroa. Over in the hexagonal pattern is Apis dorsata and Florea. And you can see, really, and there's also other, depending on who you talk to, there's 10 to 12 to even less than that species within the genus Apis. It depends if you're a lumber or splitter. But you can see how most of the species within the genus Apis overlap. They share a range. Whereas Apis mellifera is completely allopatric or separated. And the vast range that they have, that is amazing. I show people who study other organisms, like some of my wildlife colleagues, this kind of distribution, and their jaw just drops by the amount of different ecologies and climates that Apis mellifera has successfully been able to evolve and adapt in. What a diverse insect. The genetic toolkit within Apis mellifera is just fantastic. And that's really allowed us to move them everywhere and allow them to be successful everywhere. All right, so I don't expect you to read all the little marks on here. This is the subspecies map. Because Apis mellifera is so diverse and it has such an expansive range, actually scientists have used morphological characters, so wing length, wing angles, hairs, hamuli, which are the little hooks that hook the wings together when they fly, tongue length, all of these suite of morphological characters to actually put them into different subspecies. And right now, there's over 24 different subspecies that have been designated using these morphological techniques. And that's really useful for us as beekeepers, bee enthusiasts, and bee scientists, because now when you say, oh, the bee's down here, I can just say Apis mellifera scutellata, and you know exactly a little bit about the behavior, what they look like. It allows us to have a common kind of talk, lingo about these bees, which is really, really nice. And what is actually truly amazing is the fact that now, in the age of molecular techniques and all this advanced technology, when we actually look at the mitochondrial DNA, the maternally inherited DNA of these bees, they support the morphological designations that were started in the 1950s. I guess hair on my back just rises. I just love this. So cool. So you have the M lineage up here. This is Apis mellifera mellifera. And if you look back into the literature, Rudner, who did a lot of the early morphological work, actually grouped them into these same similar lineages or evolutionary branches, depending if you're looking at the DNA or the morphological features. But the fact is that they support each other, which is fabulous for us as scientists. So if we bring them down, we have the M lineage, Apis mellifera mellifera. We have the C lineage, which contains a bunch of different subspecies. Ones that are of interest to you would be the Italians and the Carniolans. And then also the A lineage, which contains, oh my goodness, I can't even go through them all. There are a lot of different subspecies within the A lineage. It's a very, very diverse area for honeybees. And that's because of the variation in climates and ecologies in that region. And then the O lineage, which I'm really not going to discuss too much today, other than kind of show you this chart based on information that we know, showing the different subspecies that we know have been brought in, their origin, their approximate arrival date into North America, and then that kind of evolutionary history, what lineage they belong to, so that ancestral piece. So mellifera, the Italians, Carniolans kind of lineage, African, or this Oriental or O lineage, which the reason I don't spend too much time on it is because it's still kind of a quagmire right now, because they're constantly getting more and more information in about, it's not an area that's easy to go and sample because of just unrest in the area, and so there still needs to be a lot more work done on the O lineage, but it's fascinating. All right, so these ones here that I have a pink sun next to are actually the subspecies that our stock has been derived from, mostly. And of course, there's other bits and pieces, and we'll kind of go through that today. But these are the Italians, the Carniolans, and the Caucasians. And really, I started my work first as a beekeeper, and all the way when I started my master's at Lynn, there were problems. The queen producers were having problems with their queens, with mating, and so this was back in 2000, and I think that's when I started my master's. I don't remember. It all was a blur. But I'm saying it was before CCD. There was problems in the industry, and so we were looking at different possible causes. And in 2006 and 2007, when CCD kind of hit the media and went mainstream, it really did a wonderful thing. Even though I don't like a lot of media drama, it really started to educate the public. And it bring up the conversation of what's going on and how important pollinators are. And, you see these survey accounts, since then, this 30% annual loss. And you read, oh, another weird virus was found. This one's a DNA virus. This is a reverse transcriptase mutato-virus. They all have these different names, and they just keep compounding and coming. Yeah.
Just a quick clarification. People talk about CCD as being a disappearing bee thing, and it not being a dead out. So you find all your bees dead in the bottom of the hive. That's not colony collapse disorder according to some people. I'm just looking at that picture and I'm just, is that correct? Or is
Absolutely. CCD is a symptomology. It is, it's weird because it's defined by the symptoms. Okay. And so it's the presence of, and you can look it up, it's like the symptoms of CCD. The queen's still there, there might be a few workers around, but otherwise the workers are gone, there's a bunch of untended brood, and nothing will rob out what's there. So basically the workers have gone. This is actually a picture of a pesticide kill in my But I'm just saying, this just represents honeybee decline, because CCD, I think, is a really small part of the problems with honeybees, personally. And actually, we're talking to people who work on the cap grant with CCD. I think they're realizing that too. It's a lot of problems.
Yeah.
But that's a good question. All right. So one thing in response to these honeybee declines, Dr. Steve Shepard was really interested in looking at genetic variation. Genetic variation is a buzz term in a lot of biological and ecological studies because you can actually learn a lot from assessing the genetic variation of a species. And there's been some wonderful studies done actually looking at genetic variation at the colony level in honeybees and the wonderful benefits that this intercolonial genetic diversity provides for the honeybee colony. Some of that work was done with Tom and Heather Matala, his student, actually looking at differences in colonies that were reigned over by a queen who was quite promiscuous, had a lot of dates, versus queens that were, not promiscuous. They maybe only had one drone pair or father. And they found differences. I'm just kind of going to lump it up because I have a lot of stuff to talk about. But they found in hives or colonies that were more intra-colony at the colony level diverse, that there was more efficient transfer of information in regards to foraging resources. They would forage for longer. So there was an impact on their foraging behavior. Also, pest and pathogen loads. There's a difference in pest and pathogen loads and how those bees responded to pests and pathogens. Well, not the load, but how they responded to the pests and pathogens. Also in the microbiome and the microbial gut flora. And there's more beneficial microbes in colonies that are more diverse at the colony level. And actually more negative or malignant bacteria in colonies that are less diverse. So there's a lot of research supporting this idea of genetic diversity at the colony level. But what about at the population level? And so a lot of other studies look at population level diversity measures. And they're important because you can establish sustainable management and conservation practices. It helps explain species dispersal and relationships. So you can look at gene flow and how kind of genetics and genes travel across the landscape. And also, variation is correlated with increased longevity and health in other species and in honeybees at this colony level. So there's a lot of benefits. And also, if we just go back down to the basics, artificial selection breeding has been going on for centuries and centuries. And that's all this is. Differences in behaviors and traits is genetic variability. And that is what breeders use as the raw material for selection. And that's what they use to make these different lines that have these different traits. So what is the variation in honeybees? Well, I like to start off by completely abusing the word population. So get ready. I'm going to simplify it out of its scientific context to commercial populations and feral populations here in the United States. And commercial populations, by my definition, is basically controlled by two different main areas, the western commercial breeding population and the southern or southeastern commercial breeding population. Yes, there's been more and more queen breeders pop up since then, but really, if you look back in the history and you map them all out, there's been two main queen rearing hubs throughout for a very long time, for centuries in some cases, especially in the southeast. These queen, and it's not just, they don't just breed bees, they breed quail, they breed coonhounds. I mean, they're breeders. And their daddy did it, and their daddy did it, and so this is like a family business that they've been doing for long periods of time. And you really can find some interesting stock in these areas too, because this is stock that their daddies probably collected in some instances right out of the woods. And then we have these feral populations, and these are represented by any colonies or nests that are not maintained by beekeepers. And I do like to, for the purpose of my talk, separate it out and say that feral colonies in this talk are anything that was collected before Varroa really became established. That's just how I'm using it in this talk, okay? And anything after that I'm referring to as unmanaged, because we don't know. Because what I'm going to show you is that the feral bees, the ones really before Varroa kind of took its toll, were genetically unique. All right, so some of the things that we were concerned about are things that possibly could affect the genetic diversity at the population level for honeybees. was the introduction of small founder populations during the early settlement of North America. And if you look back into some work that Steve Shepard's done a lot of work on this, and this is actually a quote from him. If you look and think of, there's more than 24 subspecies of Apis mellifera, and we had about 7 or 9 in total brought in. That's about 1/3 of the total subspecies. So we've reduced the overall diversity right there. So there's your first potential bottleneck. That one never really bothered me so much because actually, if you think about it, we brought quite a few in, and they were allowed to mate together in areas where they would be isolated, not really able to breed in their natural area. However, what bothers me about this is the fact that of those 9 that we brought in, probably only very few actually made it across that ship's journey and were the progenitors for the large populations that we have. If you look back and go into the journal entries, it'll be like, oh, 10 were brought over, 1 made it. You know, so the actual success of them being able to get them all the way over here across that ocean and then have them established over winter for that first season was very low. And so that, I think, is more troublesome and really created a bottleneck. And this is just a description of a founder event or a bottleneck. If you think about the initial population that they had to choose from, say we take that first kind of importation of the one that we know about, the German darkbee. We were to just grab a few colonies and say that's the variation there. We were to grab that, bring it over, and then out of these, maybe just that one made it. You know, that one actually survived. And so this would just be a square showing just that one bead over here in the left-hand corner. That would be the variation that kind of started proliferating and swarming and going into bee trees through the forest. So you can kind of see how quickly things can bottom. All right, and the second one is the overall reduction of feral and commercial populations by Varroa. We know that Varroa really reduced the population. This is probably one of the most significant bottlenecks that the honeybee population has gone through here in North America, and it's still kind of working its way through. And then finally, the use of the small number of queen mothers to produce annual replacement queens for managed colonies in the U.S. Though these numbers I'm about to show you, they're dated, and so this is a snapshot into what the queen rearing numbers were for 1993 and 1994, and 2004, 2005. So I'm snapshotting those 2 time periods. Things I can't say what is going on today, and just thank goodness bees are very, very good. Their whole reproductive biology is designed to keep variation in play, because some of these figures are pretty shocking. All right, so in 1993 and 1994, when these were sampled, workers were sampled from all the queen mothers used from all of the different queen breeders in these 2 areas. And what we found is that 603 queen mothers were used to produce replacement queens for about 1/3 of all managed colonies in the United States. Because there was about 3 million registered beekeepers during this time. And in 2004, 2005, when we repeated this study, we actually found that the number of queen mothers used actually decreased, but they were still being used to produce about almost 1 million replacement queens as well. So you can just understand another bottleneck that's occurring here. But like I said, when we look at the results and see if there was a loss in diversity, it's just amazing how hardy bees are and how robust and how their reproductive biology actually ensures outbreeding and maintaining that diversity that's so important to them. All right, so this figure here, I really like pie charts. I know people who can't stand them, so I apologize if you're one of them. I think I just love pie, so but these pie charts here are based on that mitochondrial DNA, looking at the maternal ancestry of the bees. So those same bees that we took and sampled from the commercial populations in 1994 in the West and 2004 in the West, we also looked at the maternal ancestry of those bees. We just wanted to see if we could capture genetic diversity at that level, at the maternal ancestry, kind of the roots. And what we found is that most of this stock really was the C lineage, this Carniolan and Ligustica, which is exactly what they're telling you they're selling. So I was like, oh, that's pretty honest. That's pretty good. You can see there is a loss. There is a loss of this M and a reduction reduction in the C3, which is another strain within this kind of C lineage. So there is some loss, but it's not significant. Overall, these other two, the C1 and C2 haplotype, are dominating the stock. Now, if we look at the southeast in 1993 and 2005, you can see there's a little bit more diversity. Still, it's a small chunk of pie, but you have more influence from Apis mellifera mellifera. We have more different, we have different haplotypes we have M7, M7 prime, and M4 prime, and these are all different variations and types of Apis mellifera mellifera that we can trace back to different origins. And so there's more influence because they were brought over 200 years prior to when other bees were brought in. So they had time to proliferate and kind of expand into tree cavities and flourish here. And then you can see in 2005, you also see a reduction in that small piece of pie, although it's not significant really because it's so dominated by the C lineage stock. And also you can see the presence of a new thing, which is A. So we see actually one of the breeder queens actually had an African maternal ancestry. So you can see the influence of Africanized bees as well.
Do we have any more current data? Like is the bee team going around getting like 2013 figures? Or?
You know, they probably are. I would talk to Steve about that because
he's 7 years.
I think, you know, 7, and I did a 10-year span, and I don't think that's really long enough, honestly, to see any gigantic change. But it would be interesting. I hope he keeps it going, and maybe every 10 years just kind of checks it out and sees what the change has been.
You said Apis mellifera and mellifera. So is there a difference between the two?
Apis mellifera is the general species, the binomial. Apis mellifera mellifera, when you add that third name, is the trinomial, so that's the subspecies. So there's Apis mellifera mellifera, which is the dark German bee. There's Apis mellifera ligustica, which is the Italian bee. So they're all Apis mellifera, but within Apis mellifera, all these different subspecies. So there's variation. And I always think of it like Philadelphia, because that's where I originated myself, and I was just like, well, Southwest Philly is mostly Irish, you know, and we're all American, you know, humans here, but South Philly is Italian, you know, so it's kind of like that. If that's a really bad analogy. But, all right.
But what I'm really asking is that would you categorize like the colonies or whatever there is out there to be, say, Apis mellifera, not sure, and then say buckfairs or Russian as
They're all
mellifera.
They're all Apis mellifera.
Oh, they're all Apis.
They're all Apis mellifera.
Oh, okay.
And when you get into buckfast and say, I think one of those was Midnight and Starline, those are more like stock that was made, like hybrids and things that were selected and constructed. These are really subspecies that were designated based on morphology. So it's a little bit different, but it is blurry. All of it is, there's fuzzy lines between it all. All right, so also we had samples in bees, about 692 feral nests that were in the freezer, collected between 1980 and 1992. And if you look at some of the charts, it's really 1992, actually 1993, where you start to see the drop from Varroa. So this is really before Varroa kind of was established and really taking it hold. And so this is just such a neat thing to have access to, the sampling. And what you can see here is, again, it's mostly dominated by the C lineage. But what's so interesting is this presence of Apis mellifera mellifera. You're really starting to see that there's a whole lot of bees present at the same time that are Apis that have this different ancestry, very different ancestry. So also we're seeing Apis mellifera lamarckii. It was brought in 1866, I think, and we're still seeing pockets of it between 1980 and 1992. And actually, some recent samplings I did, I'm still finding pockets. Of course, this is maternal ancestry. So the nuclear DNA might not look anything like, isn't anything like the original Apis mellifera lamarckii, but you still can trace the maternal ancestry, which I think is really neat.
How were you able to?
Excuse me?
How were you able to trace it back? to
By
like,
looking at their mitochondrial DNA. Because the mitochondrial DNA, you as well as these bees, you get it from your mom. And she got it from her mom. So you can just trace back maternal ancestry by looking at specific areas, in this case, in the bee's mitochondrial DNA, and be able to tell where they came from.
Deborah, I have a real quick question, I'm sorry. When, and maybe you mentioned this and I missed it, those feral populations, the samplings that you guys had in the freezer, did they come from just a specific area or kind of across the nation? Or?
No, and actually, because I have a really bad memory, I have a slide that shows the different states they're from. They're from 11 different states.
Okay.
And I'll just point it out to you once it comes up, because I'm going to mess them up if I tell you.
Thank you.
Sure. All right, so that was just the mitochondrial DNA, looking at paternal ancestry. But we know that's interesting historically, right, if you're in family trees. Well, the real nuts and bolts is if we look at the nuclear DNA, because that's where, we have behaviors, it's from mom and dad, from drone and queen. That's kind of more of an interesting thing. And when we looked at 10 different loci on all these bees, the same ones, and compared them to see if they shared alleles or if they had different ones, or to see if they lost them over that 10-year period, we found that there was a significant loss of allelic diversity. And I want to say, but, and Steve Shepard tell him, but there was a significant gain in alleles from 1993 to 2005. So there wasn't a loss. Okay, that's the overall happy message from this, is that we didn't see a loss in diversity over that time period. Was there a loss of the old alleles, a significant loss? Yes, but we got new ones. So that's a good thing. And so I was asking the audience yesterday, what could be some possible contributions? Where could we have gotten these new alleles? Anyone shout it out?
Well, from like the mixing of subspecies that never interacted before.
Possibly, and maybe some other things have been in the news. Africanized bees, as well as what was something else that was brought in during that time period.
oh, the sperm from Sweden.
Dark little bees.
Russian bees.
Bees. So we did, and there's probably other things as well, but we have known things that were brought in that could have supplied unique and new alleles and to add to diversity. All right, so this is something we wanted to know if the Western commercial breeding population and the Southern commercial breeding population, they're pretty isolated geographically. And we wanted to know if they were different from one another. So we put it into this awesome program, which I love very much. And based on all those allele frequencies of that nuclear DNA, we found that actually they are different from each other, very different from one another. And so this is good because they could potentially use each other's stock as a source of diversity. And there's been a lot more movement, so it would be interesting to look at this again as well between the two groups. And there's a lot more queen breeders and queen rearers now too. So it'd be interesting to kind of get a more total sampling. But you can see also, this is the point about the 10-year time frame. Over 10 years, there's very little difference within the region. There was very little difference. All right, so this is the most exciting part. This is where I actually have the states listed. So there's North Carolina, and this is the feral bees. So they were sampled from North Carolina, Arizona, Louisiana, Georgia, Alabama, Mississippi, New Mexico, and South Carolina. And they were actually compared, their genetics, to all of the commercial bees that we looked at. And over both sampling periods, they were just completely different. They're just completely different genetic structure. And what's so exciting about this is that they coexist though, so they're near each other, yet there's very little gene flow occurring. And they've existed together for a very long period of time, and yet you still see there's some barrier to genetic flow. And so there's lots of wonderful ideas of what mechanisms could kind of be behind this. And a lot of them have to do with behavior of the different bees and their usage probably of drone congregation areas and the phenology of when they use these drone congregation areas. So there's wonderful things to test. I didn't do any of that, so just wonder. But there's some really interesting other people's work that have looked at it in France with the Lalande, with the bees, the prototype there on heather, and they actually have shown that those bees actually use the drone congregation areas at different times than the commercial bees in the area, bees that were brought in. So you have this reproductive barrier in that instance, but I don't know what has maintained this. All right, so now I switch gears a little bit and talk about the loss of the feral population. And you probably have all seen the statistic, the status of feral honeybee populations in the U.S. remains unknown after 90 to 95% reduction. Well, I haven't yet found the paper to prove to me, to show me the numbers that there was a 90 to 95% reduction of the feral population by Varroa. If anyone has found that, I have to admit, I do get bored when I'm searching through articles. Please share it with me. I have not found it. And so once I saw those differences, it really brought up the question, does the unique identity that we found of those feral bees, does that still remain? Because I was getting calls, oh, there's a bee tree here. Oh, these bees have been in this church for 50 years. Just all of these different calls where you come and remove these bees, you know. And so what are they? That was my curiosity. What are in these trees? What are in these different structures not being managed by anything? So I got a grant to actually start the Feral Bee Project. And because I'm just one person, I decided to enlist citizen scientists. And so basically I gave talks all about this to kind of promote it, get people involved, get the word out. And basically anyone can still go on to SaveTheHives.com and put in the coordinates of an unmanaged bee structure, be it a tree or whatever. There is a filter there because back in the day, I used to literally drive days to get to some of these, and if I get there and it's a yellow jacket nest, I'm going to give you bean calls. So I ended up putting a filter in so that I could make sure. But it's weird because I did find a lot of very interesting in-ground honeybee nests. So yeah, but you can see here, these are just some of the locations. I started this project when I was in North Carolina, so the map you're seeing right now are bee trees and bee structures that are mostly prevalent in North Carolina area. And one thing that we've done so far is we actually hooked up with Dr. DeYoung in Brazil, who does this geometric wing analysis, which is a different type of morphometric analysis that's very accurate. And we had him look at our bees. And so the blue circles and the red squares and the green diamonds are all bees that I have sampled. And so the natural environment means those ones that I'm talking about, the unmanaged bees. And these are ones that I collected in North Carolina. The managed here, the managed Lewis, Colesville, and Jay Lee, right underneath the natural environment, are managed bees that were collected in the same area in North Carolina. And as you can see, based on morphology, they're lumping together, suggesting that there is a lot of escape swarms, or there's similarities in their morphology when we're just looking at the morphology, the morphometrics. Actually, this is the interesting thing to me. He actually also compared those to the species type for Apis mellifera carnica, Apis mellifera caucasica, Apis mellifera ligustica, and Apis mellifera mellifera, and they completely separate out, which you would expect after being removed and interbreeding for a long period of time. But this kind of supports this whole idea that a lot of the bees we have here, they're just mutts. They're mutts that aren't anything like the true subspecies that where they originated from. And so when people say I have Italians, I can say, yes, you have very nice yellow bees. Right? Or it's likely, you know. And it's not to say that their breeding efforts are bad, but in terms of saying that you have Apis mellifera ligustica queens or Italians or carniolans, I think that's a little far-fetched at this point. All right, and so now I'm just going to briefly talk about some work that I was just doing the genetics for some work that Tom Seeley's conducting in the Arnot forest, and you can see his contraption here. I'm still just amazed by your procedure. His beelining is amazing. And you see in some of these pictures, if you were at his talks, which I hope you were, this little person here, and the entrance to the bee tree very, very much up at the top. And this Arnot forest is really amazing because it's this kind of wooded area, this large wooded area, but it's also then surrounded by forests. So it really is this kind of isolated bee forest paradise that luckily Tom had censused 3 times now, 1978, 2002, and again in 2011. And the coolest part about that is that he always finds lots of bee trees. And the extra cool part is they have mites. So they actually haven't reduced in number at all. And he hasn't even censused certain parts of it. So there's probably even more than he or anybody else knows. Yet, in spite of all the problems going on with honeybees, they are just maintaining. And they have one of the worst enemies to honeybees, as far as I'm concerned, the Varroa mite. All right, so from there, this is also part of the feral bee project in a sense, and that this is, I really wanted to look at a natural population, and this is the closest thing that I know of in the United States to a natural, or what we know of as a natural population where the bees are determining their density based on their parameters. They're making the decisions without any input from us. So this is really exciting. So in 2011, in late August, Tom did his magic and found lots of bee trees and sent me lots of dead bees. I know that was probably hard for you, Tom. He does not like to kill them, and I understand. But for the sake of science, we pickled them, and he sent them on to me. And I also want to say that he was very thorough in that he actually looked for any managed apiaries surrounding the forest. And so the first apiary was about 0.5 miles, and he sampled all the colonies from them and sent me bees from each of those, as well as the second apiary. He sampled all the hives from there and gave me bees from those. And what we did was very similar to work that I've done previously, where we actually looked at the nuclear DNA. And what we wanted to see is, are these Arnot bees, are the genetics different? Do they have a different genetic makeup or toolkit than, say, these managed bees in the closest surrounding apiaries? And what we're finding is similar numbers, just extremely different. So what does that mean? I don't know, beyond that the genetics are different. They have a different genetic makeup between the managed bee yards and the bee trees. Also, what was interesting to me in looking at this, having worked with genetic diversity in honeybees, is that there's a lot of different proxies for genetic variation and genetic levels in these bees. And the allelic richness was pretty similar among the 3 groups, the tree bees, the BR1 and BR2, which is the total number of alleles that they have per area that we're looking at on the chromosome, okay? But what's really cool is the private alleles. These are alleles that are completely unique. And the mean here is for the private alleles for the tree bees is super high and completely dominates, is significantly higher than BR1 and BR2. So that's really interesting. What are those private alleles? What do those unique genes refer to? So that's kind of really interesting. All right, and so then we put this genetic information into a program that establishes how many populations you have based on your genetic data. And so it determines K, and K is the number of populations. And as you can see here, we have 3 different populations. We have the Arnot forest bees, or tree bees. We have BR1 and BR2. And what's so wonderful about this program is it can determine levels of, or how genes are flowing, so introgression. And so what you're seeing here in the Arnot forest bees, you just have very, you can't even say that there's introgression. There's like little tiny yellow blips. But what we're seeing in BR1 a little bit and BR2 is that the forest bees are genetically, there's gene flow a little bit more from the forest bees into the BR bees, BR2. But there's very little influence from the managed bees into the forest bees. And Tom's alluded to possible reasons because of this, just sheer numbers.
That's interesting because BR2 is farther away.
Absolutely. But if you think about it, generally a lot of the literature supports that bees will fly to further drone congregation areas. They are wonderful at trying to outsource to keep their variation. That's just one thing that's really amazing. So this makes even more sense.
Just a comment on that, that's very perceptive. BR1 was established in 2011 from Queens, purchased from Mellifera in Northern California. So BR2 had been there for many years. It's had more time to pick up genes from the rest.
And that too. Yeah, now I'm glad I know that now. All right, and so this is something that is in works right now. So if you're familiar with reproductive biology of the honeybee, we have the queen right here. Those little lines with the different colors is a very simplified idea of her flying around with her sex locust flying, her free flying there. And these are all drones. We know that drones are haploid and that all females are diploid. So they only have one sex locus. And you can see there's variation. This is a pretty diverse drone comet chasing this queen right here. If they were to all mate with her, she would hold their sperm in her spermatheca, each representing this diverse amount of sex loci here. And her offspring will contain one sex locus from her and one from the one of these drones, if it indeed becomes a viable worker female. And so by looking at a certain number of females from a colony, or from a bee tree, we can actually determine the queen's genotype, as well as how many different drones she mated with, or patrolines within that bee tree, or subfamilies in that bee tree. So we can see if the intra-colony diversity is greater in the bee trees than it is in the managed bees. And maybe that, since intra-colony level diversity has been shown to have such wonderful qualities and impacts on bee health, if that maybe could be a reason why these are not forest bees. So that's something that's in the works right now, and we should have data fairly soon, because our collaborator, David Tarpey, is a lot faster than me at analyzing the data. So that's to come. All right, so I'm going to switch gears before lunch and talk about this new project that I'm doing, because I'm really excited about it, and I'm hoping that the West Coast will come on board when we do our next phase, which is getting collaborators, beekeeper collaborators. We really want this to be something that is driven by beekeepers and that's usable to beekeepers. So that's kind of our impetus. And of course, I'm going to focus on the Varroa mite really quick here. I'm not going to go into too much detail because we know about its nasty little habits. We also know that it's native to Southeast Asia. Its true host is Apis cerana. It has two distinct phases, this reproductive and phoretic stage. And it can hide sometimes, but other times it's just like, look at me, you can't get rid of me. It'll be just like right on the thorax. It's so annoying. So this map here shows kind of the spread of Varroa from 1912 up to, I forget what date, in the 90s somewhere. But it's really interesting to watch how Varroa kind of goes, through this time flow, all the way to, and how quickly they just kind of spread across an area. It's just, I love watching this. It's just kind of amazing to me. What a tremendously successful pest. And you know why? It's because we were so good at moving them everywhere. We are really to blame for this rapid dispersal. All right, so I think that's it. It just goes up to 1998. And for those of you who are interested, there's a lot of history and speculation of why and who brought them into different places in, say, Wisconsin and Florida. All right, so the life cycle. So what's the difference? If you think about the true host, Apis cerana, and Apis mellifera, what are the differences? And if we look at the life cycle, overall we can see that Apis mellifera, the workers and the drones, have a longer capping. They have a longer capping period. Versus Apis cerana is about 11 days capped. Well, the foundress mite will enter the cell shortly after that capping period. All right, and once she does, about 60 hours after capping the cell, the Varroa begins to oviposit. And of course, the first thing that the first little critters she oviposits are little males that she very much is incestuous with, and then which produces the females later on. If you look, generally it's 2.6 female offspring per drone cell and 1.3 female offspring per worker cell, but this does vary depending on where you are. But this is just some general background. If we also look at some differences, in Apis mellifera, Varroa reproduce in worker and drone group. So that's a bummer. And in Apis cerana, Varroa reproduce solely in drone group. They have shorter cap developmental period than workers, and so it doesn't really allow for the Varroa mite to reproduce. Also, in Apis cerana, you have different behavior. You have much more hygienic behavior. being exhibited. There's worker recognition of Varroa. They can detect and remove diseased brood. Also, there's more auto and allo grooming that occurs. And also, there's been a lot of research into fluctuations on age and the mites being able to detect those different fluctuations and how those affect the mites and their reproductive potential. So there's a lot of interesting relationships going on.
What's JH?
JH is juvenile hormone, and that's a really important hormone in all insects, but there's actually different types of JH, and it's involved in molting, going from the larval to the pupal to the adult stage. It's involved in way more things than I think we know. All right, and so if we look at the population dynamics, and this is something that's very generalized. It probably might be a little bit different depending on where you live, but overall, I think this covers it. If we look at the bees, you can see that basically around April and May, once that nectar and pollen flow really start, the population just starts to shoot, and that's because you have this increase in brood. You have this kind of feedback loop between the bees bringing this wonderful stuff in and the queen, starting to lay eggs. It's just this feedback loop. And what the bummer is if we look at mites, they're slowly rising as that brood increases. But as soon as they're at their highest level is when the bees and the brood population start to decrease. And that's right when you have your winter bees going into winter. Perfect timing for the little critters, right? And so that's when you see a lot of crashing, at least in my area. All right, so another awful thing about Varroa is that they vector viruses, a lot of viruses. And the problem here is that generally these viruses don't have a lot of overt signs, except deformed wing virus is pretty obvious. But even still, when you see a lot of deformed wing virus, at this time, it may be too late. So these viruses are really a problem. And there's a lot of tools that have been developed to help beekeepers actually deal with these mites. There's Apistan, check mite. There's also additional miticides like Mite Away, Apilyte Var, Apivar, Apigard. If they do any more Apa something, I'm going to freak out because I can't even keep them. I can't keep track of which is which. And then HopGuard. So there's a lot of different things that you can put in the hive that actually can help reduce mite loads. But there's also problems with that, which we'll discuss. There's also genetic tools. And these are, different people actually selecting for desirable traits, one of them being disease and pest resistance, or certain types of behaviors that help detect disease and pests in the hive, honey production, overwintering, swarming behavior. These are all different tools that we have available to us as beekeepers and options that we can kind of think about and ponder. There's also monitoring techniques. So we can actually follow and keep track of the health of our bees as well as the presence of pests. And of course, there's mite monitoring techniques like sticky boards, powdered sugar shakes. There's also things where you can do purple-eyed poopy scratching, where you scratch them all out and can count the number of the reproductive. You can really get an idea of the reproductive potential of the mites when you do that, but it's a pain. And so there's a lot of different things that we have to follow depending on what types of treatments we use. And it's really tough for beginner beekeepers to digest all this and know what to do. Think about it. And, when I first got into beekeeping, it was like, oh, honey, I forget who said it yesterday. It was perfect, though. Like, oh, the bees. Like, those are the things you want to be thinking about. Not like, oh, I shouldn't treat here. What should I treat? Oh, it might have toxic effects. Oh, it might blow up my queen. Those are not things a beginner should have to worry about. So, problems are grower building up a resistance and to all these chemicals, and they build it up quickly. And I think that has a lot to do with the density of our apiaries, that these mites actually, the drifting, and so the spread of resistance actually really happens quicker than it might. Also, some of these things are temperature sensitive. They can blow up your colony. I've blown up my colony before with some of these treatments in the past. Queen and drone development. We've seen studies showing that there's effects on queen and drone development. That's awful. There's residue problems. I'm just waiting for them, all of the studies so far showing Chinese honey as evil. I'm just waiting for that study to say, U.S. honey, this found in it. There goes our honey market. That would be, I'm a honey pig. That's why I got into beekeeping, really. If that happened, I'm going to just cry. Comedic substances, pheromone disruption with some of these more essential oil-based. Bees communicate via pheromones. via smells. When you put that eucalypton thymol in there, they're completely disoriented. I've seen it in my colonies. Maybe you haven't. Maybe it works better here. In Delaware, not so good. I can't even think. I'm like, oh, it smells like a giant cough drop, my whole apiary. So I have issues with a lot of these, and I know a lot of other beekeepers do too. Anything that you're putting in a hive to kill an arthropod on an arthropod, you have to be skeptical of or really research it to understand it, right? So we decided to think about producing this IPM approach. And of course, the granting agencies love IPM when you use that term. So IPM really originated to reduce chemical residues and to really reduce the effects of chemicals on natural enemies and other beneficial insects, but it didn't mean eradicating chemicals. Chemicals were a part of the toolbox. In this case, I'm removing the chemicals. And so this is a Varroa IPM program that we're developing that includes stock, very, very premeditated, intentional stock selection, drone brood removal techniques, as well as the use of colony fission as a tool. And we also use screen bottom boards. My students always like, put the screen bottom boards in there. We do that too. I'm running over, so let me just get through and then we can talk later.
What's fission?
Huh?
What's colony fission?
We'll talk about colony
Okay.
fission in a minute. That's just a fancy name for splitting and swarming.
Okay.
We'll talk about it. So the objective is to develop an IPM program for small-scale beekeepers, which are the main people in my region, that will help alleviate costs and detrimental exposure of bees to in-hive miticides. We're doing a survey. We've actually sent out 300 emails to different beekeepers in our area, small-scale beekeepers. We've had 294, as far as when I left, respondents. So this is important to people, because if it's not, they won't fill it out. And we're getting inundated with emails. Beekeepers in my area are craving something, some other way to manage pests in their hives. All right, this is a survey link which I can give you guys later if you're interested to be, join in on the survey. This is just a little bit of information about the beekeepers in the Mid-Atlantic. The numbers, there's about 11,000 throughout the Mid-Atlantic area. The amount of colonies, you can see here there's differences in summer and winter because there is migratory beekeepers that come through. And also, the IPM approach is really, like I said, stock selection, trapping, and brood interruption. And if we look at this IPM triangle, you can see here, this is your classic IPM triangle. Generally at the bottom, it's more preventative techniques, cultural control, physical and mechanical. And as you go up this pyramid, you're going up into intervention and more toxic, more immediate, like emergency measures. Because of course nobody wants their colony to die, and sometimes you need to go in there and do something like me when I'm just like, I don't want to feed, but if it's the difference between a dead, starved colony and a live colony and me giving them a little sucrose, I'm going to feed my colony, of course. But these are choices we all have to make. It's a personal decision. One thing that we're using in terms of our stock selection, and this might be different for different people, which we'd like to track, is the use of Varroa-sensitive hygiene stock. And so we've actually got bees from a breeder with the Varroa-sensitive hygiene as one of their selective criteria. We've been monitoring the trait, and we do see the trait actually in all of the colonies, and they're wonderful. And what's really interesting is with this Varroa-sensitive hygiene, we've taken pictures. This is when I was in England, and you can see pieces of the honeybee pupae, like their antennae, on the bottom boards. It's just covered with pupal parts of the bee because they're dragging them out as soon as and just bit mites with bite marks in it. And I know there's breeding programs that are going on for breeding mite biter bees. Bees are just like, they see a mite. So that's pretty neat. And one thing people get disturbed about first with VSH is that, I have a shoddy, shotgun brood pattern. And actually, we have to kind of change that a little bit because if you have Varroa-sensitive hygiene behavior in your hive, you're going to have what looks like a shotgun brood pattern, but that's the bees pulling out those pupae. So at first I was wondering, why is it so spotty here? But it's actually the bees pulling them out. And then you can see them actually, once the queen lays in it again, you'll see the ridge where they uncovered it originally and pulled the pupae out, and then there'll be a thin layer of wax in the center of that, so you see a ridge. So you can tell which ones they pulled out. It's really, really interesting to watch this behavior. There's videos that you can see the behavior in action if you want to. All right. So the drone brood removal, this is a technique that has been studied and shown not to have long-term effects on Varroa levels. However, I think it can be useful in combination. And also, for our study purposes, we are freezing the drones, but I definitely, for when I'm writing up the IPM program, I'm going to be talking about having a sacrificial colony in another location so you don't kill off the drones. It's really important to conserve our drones, especially any drones that might be able to deal with the mites. So by freezing them all, you're not giving them a chance at all. And you're losing out on variation, and so it's a real problem. So for ours, we have to freeze it so we can count the number of mites in the cells, but for the overall IPM program, we're not going to be doing that. All right, now quickly, colony fission is the next one, and I'm going to talk about natural, which is swarming, which this is probably not 50% workers. You should ask Tom how many actually. And the parental queen will actually leave and make a daughter colony in, say, a tree or somewhere, leaving back the parental colony here with a virgin queen. And so this is the idea of swarming. This is how they reproduce at the colony level through the landscape. It's a fascinating, exciting, amazing thing. And also then splitting, which really is artificial swarming that is beekeeper induced, where we try to mimic swarming. And swarming has been shown to actually, in a lot of instances, be possibly, especially in other countries, this work has been shown or done more, an evolutionary technique that reduces pests and disease within honeybee populations. And if we look more into it, what you have here is brood interruption. And so that's the nuts and bolts of this. It's a break in the brood cycle. The new queen must be reared in the parental colony, depending on the weather and the alignment of the stars. It could be 2 to 6 weeks. In the daughter colony, the comb must be drawn. There's other factors before that queen can begin laying. So this break in the brood cycle also breaks the brood cycle for Varroa. So this is a very interesting tool that can be used. The main problem that I've heard from beekeepers is that split going to survive, right? So if you can do it early enough on and be kind of premeditated and intentional about it, that's what we're looking at to still see if you can be productive in your apiary. And we'll also talk about other ways you can be productive in your apiary still using this technique. All right, so we have actually 2 different apiaries doing the same project. Penn State University as well as University of Delaware, that's our research apiary there. And this is our design. We have 24 original colonies of the same stock. We've divided them into 12 control and 12 treatment. The control colonies are here, which will be colonies that are prevented from swarming. And then we have the 24, the 12, which then we split. And so that leads to 24. And then within the ones that we split, we have early spring drone frame removal. So we're testing if early on, if we just do 1 drone frame removal, what kind of effect does that have? Versus monthly drone frame removal, which can be laborious depending on how many colonies you have, and then neither. And so those are our treatments within there. And we're also, this is the most exciting part to me, my teaching apiary, which is pictured right there, is surrounded by tall trees. And the swarms, there's nothing else really around, so the swarms go straight up. So I have a 90 to 95% recapture rate of my swarms, which is great. So what we've done early in the season is we've marked all of the queen's different colors. And we now have captured, not as many as I would like, I didn't have as many swarms for some reason. We have 6 swarms that we've caught and we're tracking the parents. And even though right now it might not be enough to compare, overall we would like to compare the difference between natural swarming and the effects of Varroa and Varroa vector viruses versus this kind of artificial swarming. So that's kind of the goal, because that's just neat. All right, so the protocol here, really quick: drone frame removal time periods, treatment colonies, control colonies, monthly recordings of treatment and control colonies' health. I just want you guys to get some food. And so we're looking at temperature and relative humidity in all of these to see how that plays in. We're looking at adult bee and brood populations, brood health, temperament, foul broods, not just American, because actually we're having issues with European foul brood a lot. Queen cells, chalk brood, and we're using 3 different mite monitoring techniques And finally, we're looking at viruses. So we're taking samples of bees and looking at 7 different viruses and 2 different nosema species to look at the differences between those as well, between these 2 treatments. This is preliminary data. I didn't have money yet, so we only had 5 colonies per treatment. And so this is from last year. We're in the middle of doing, we got money, and so we're in the middle of doing it for real this year, and we have 24 colonies. But this is just some quick stuff that I had my students send, so I can show you guys what actually convinced some of our supporters to give us money to do this project. And if we look at colony population growth in terms of bees, you can see the control here, you can see the parental, you can actually see in the dip here where we did our split, and then you can see the daughter colony here. All right, if we look at Varroa mite population, again, we have the control here through July to September, the parental, and then the daughter here, and this is in terms of mite drop. And this is basically mite counts per day, just a different visual for you guys to see based on our 5 colonies, looking at the controls with the swarm preventative tactics, the original, and the split. And so this is preliminary stuff, and we should get a real feel with the larger amount of colonies and the 2 replicates of what it's really going to be like at the end of this season. And the idea is to create this dynamic apiary, and this is how I run my teaching apiary. I have certain hives, it depends on the year. I have it divided into 3 groups. Some of them are honey producers, where swarm preventive tactics are actually employed. Other ones, I have a group that are my splitters that I split early spring. It also depends on the history of the colony and the age of the queen. And then I have others that I let swarm. And so every year they cycle through these different things. And it really helps to keep my apiary fresh. And also another important part is the stock. If you buy susceptible, disgusting, crappy stock, then your bees, you might as well just flush your money down the toilet. Be intentional about the stock of bees that you buy. I can't say that enough. I've flushed my money down the toilet too many times. All right, and so sustainable beekeeping, back to that idea. What is it? Well, the definition is conserving the ecological balance by avoiding depletion of natural resources. So there's all these kind of wishy-washy definitions, but overall, it's a good thing. And it's supposed to be self-maintaining, to be able to go on, and not run out. That's kind of how I think about it. And I think, if we really look at this idea of survivor stock and how bees are able to do it themselves and kind of the different important life history strategies that they've developed and maintained and evolved and put a lot of energy into. They do it for a reason, right? And so in France they have survivor stock they found that's treatment free now for, 2 years ago it was like 17 years, now it's 19 I guess. There's the existence of feral colonies here in the US in the Arnot forest in the presence of different pests and pathogens. And so natural selection acting on feral colonies and their parasites might have not even just fostered a stable host parasite equilibrium, but just it's worked itself out. So the trade-offs can occur and they can live simultaneously. So how do they live in nature? How does nature select that's different from us? And I'm going to end with this because I think this is so important too, and it's such an area that everybody needs to do research and demand research into, is looking at the quality of our forage and how much forage we have and what we should plant. Just because it says a pollinator mix, show me the research that says that has nutrition in it and that the bees will come to it. So I think that's the, almost done. Outreach component, this is for all of you. We are going to be having other people get involved in this and tracking their colonies and providing them with training and workshops and material. This is a pamphlet I put together for another product I did, which will be doing trifolds as well as a lot of web-based stuff about IPM program once it's finished. And I just want to acknowledge all these wonderful people who are involved and helped, also our funding sources, which have made this project possible. And that's my student who's doing all the work, so I had to show her. She's fabulous. I am so blessed with her. And that's, and another fabulous worker. And I have no time for questions, and neither do you, because we're all hungry. But I'll be around all day.